again. I hope you all had a refreshing break and are ready to continue. We start with GIT block, which is physiology-wise quite light, especially as you compare it to cardiovascular, respiratory system, renal systems that you have done last semester. This is very easy because it's not conceptual at all. There would be a little bit memorization, and pretty much the major concept that you have to understand is the difference between enzymes and hormones, so that you don't confuse them. What is enzyme and what is hormone, what is secreted where, this you will have to memorize. So there will be quite a bit of memorization in physiology, but otherwise it's not difficult at all, and I hope you all to cope well. So I would like to attract your attention to the handbook. Please take a few minutes and study it on your own, because it has many answers to your questions, including the literature that we recommend. The literature is found, so this is the outline of the course, the lectures and learn objectives that we are going to follow throughout the course. This shows the weight of continuous assessment. So 60% is pretty much everywhere is MCQs, 20% OSPI, and 15% SAQs. Recommended textbooks. The major one for physiology is Guyton and Hall textbook. For GIT, pretty much any edition that you can find, okay? Any edition of this textbook will do. Don't forget 500 questions, Guyton and Hall, and also BRS by Linda Constanza. So this is the textbooks that I'm using. This is the textbooks that we recommend, pretty much across the board for all the blocks, Guyton and Hall. I have to say that I have carefully selected all the questions that are relevant to the topic from all these resources that are available. So you may use 500 questions Guyton for yourself, or may not, but I'm posting practice questions, as you have already seen. So each topic would have a minimum of 10. I will keep adding them to the same links. So make sure you're going to the same links several times, and as usual, I recommend opening the questions before the lecture. Just open, scroll, go through them, check how I am putting the question, what is being asked, because the concepts that are tested by pretty much all the question books that I went through and picked up questions are the same that are tested for board exams. I'm not going into minor details, but just what you need for boards, whatever boards you decide to take, whether it is USMLE or Saudi boards or PLAB, British board exams, so whatever the questions are relevant for preparation to board exam. So this is it for the handbook. Couple of advices that I wanted to give you is, first of all, use AI. Use AI actively, Copilot, Gemini, whatever. Don't come home and just start reading the lecture slides, highlighting them, making your own notes after you have already made them in class. It's counterproductive. It may be fun. I mean, it's a good way to study. I'm not saying it's not a bad way to study, but you simply don't have time for this. With the load of information that you have, you don't have time for this. So study actively, which means do questions and use AI. When you come home, take the lecture, upload it to Copilot. Of course, I'm sure pretty much all of you are already working with AI, right? Who does not? No one, right? Everyone is working. So your AI already knows you, right? They know who you are, that you are first-year medical students, right? So ask them to maybe summarize lecture for you if you need, or start with ask them to give you open-ended questions based on the lecture, and then you answer. Either speak it to them, whatever, or write. I recommend writing because this is how you answer SAQs. And this is a kind of weak point. MCQs are usually better than SAQs. So start writing, type in answers, ask them to evaluate you, then proceed to MCQs and ask them to create MCQs for you, and then you do the same with MCQs. I'm using a lot of clinical cases, you will see them. You probably have already seen them. You can upload clinical case and again ask to make the questions, but use learning objectives. Make sure that AI does not lead you into clinical part, because though we are using a lot of clinical questions and a lot of clinical scenarios, clinical cases, right? We are not testing you on clinical things, right? So we are not testing you on the treatment choices, on the plan management. We are not testing you on differential diagnosis, on the steps in diagnosis. That's not your theme these days, right? So ask AI to give you questions based on the lecture learning objectives, all right? So work actively with these lectures. Don't just sit and scroll them and read them. No. Do questions, do SAQs, do MCQs, all right? So these are the learning objectives for today. And we are going to start with GI functions. So there are 5 major functions that we are talking about, and first of all is of course food processing, since we are dealing with gastrointestinal tract. Then excretion of metabolic waste, protective functions, endocrine function, and hematopoietic function. So food processing consists of 4 major mechanisms involved. So this is digestion of food with the help of enzymes, then a motor function of gastrointestinal tract, which is basically propulsion of this food, propulsive peristalsis and mixing movements, which allow to mix the chyme with digestive juices and propulse it from esophagus all the way down to the rectum. Then absorption of the products of digestion, and secretory function is the function of all the glands, secretory glands of external secretion, like salivary glands, like pancreas, gastric glands, intestinal glands. So all of these secrete certain digestive juices, and we are going to talk about the mechanisms. Finally, after absorption, there is excretion. So what is excreted? Apart from metabolic waste, drugs, toxic substances, there are also bile pigments that are excreted, which actually gives the color to feces. Protective function is because of immune barrier and bactericidal and bacteriostatic action of some of the digestive juices. So immune barrier starts from the mouth cavity, and it's first of all epithelial barrier that we see cross out the whole gastrointestinal tract. Tight epithelium does not allow the absorption of toxic substances. We see that it's very tight in the stomach, for example, in the intestines. There is a significant mucus secretion starting from the mouth cavity and all the way along the gastrointestinal tract. And mucus, as we'll talk about it later, also has a protective function, so it serves like another layer of barrier, the first layer of barrier, which also helps to neutralize some acids and salts that can be acids and alkali that can be ingested So immune system is represented heavily in the gastrointestinal tract, and GIT contains more than half of all the immune cells in the body. So we are talking about pairs, patches in the ileum. We are talking about lymphoid folliculi in the intestines. We are talking about lymph nodes in the mesentery of the guts. We are talking about specialized cells, lymphocytes, M cells, macrophages, pretty much everywhere throughout the gastrointestinal tract. Plus, it's immune globulin A secretion, and immune globulin A is present in saliva. It also helps to neutralize bacteria without developing inflammation. Bactericidal and bacteriostatic action. Bactericidal means killing bacteria, and bacteriostatic means preventing bacterial growth. Is due to the factors in saliva, and due to the gastric juice, due to the presence of hydrochloric acid in the gastric juice. Endocrine function. This is what we are going to talk about today in much detail. And we are talking about APUD system, which is abbreviation for amine precursors uptake and decarboxylation. This is the system of different cells. I have that on the footnotes of the slides later, amine precursors uptake and decarboxylation. So there are different types of cells, G cells, S cells, I cells, K cells, M cells, scattered all over the gastrointestinal tract, secreting different hormones, somatostatin, gastrin, methylene, secretin. So many different hormones are secreted not in one place, not in one gland, but in multiple places throughout the gastrointestinal tract. Finally, the last but not the least is hematopoietic function, which is secretion of intrinsic factor in the stomach, which helps to absorb vitamin B12, which is essential for absorption of vitamin B12, and ferritin deposits. And as you understand, both ferritin and vitamin B12 are essential for the secretion of, for the formation of RBCs. Any questions so far? So when we are talking about the major function of gastrointestinal tract, food processing, we say that there is digestion, and this digestion takes place both in the cavity, what we see here on the slide, and also on the brush border of the enterocytes, and we will be talking about this later in week 3. Then absorption is the transport of the products of breakdown from the cavity of gastrointestinal tract into the blood. While when we are talking about secretion, we say that the specialized cells, pretty much all the glands, which is pancreas, salivary glands, they secrete enzymes which go into the cavity of gastrointestinal tract, and they also may secrete hormones which go where? Into the blood. And that is the key difference between enzymes and hormones. Of course, we will also be talking about paracrines, which are secreted into extracellular fluid, but true hormones, and we are going to discuss which are true hormones, they are always released into the bloodstream, and with the blood, they can be carried to all the cells that have receptors to these hormones. All right. While enzymes, those that are partaking in the breakdown of proteins, fats, and lipids, they are secreted into the cavity of the gastrointestinal tract. Whether they are released from pancreas, from salivary glands, from the stomach glands, or from the liver in the form of a bile, but they go into the cavity, all right? They are normally, if we are not talking about diseases, and we will be talking about diseases later, like pancreatitis, for example, so they are not going into the blood. Enzymes are released into the cavity, true hormones are going into the blood. The gastrointestinal tract has pretty much the same structure everywhere, with certain peculiarities that we will be talking about when it comes to, for example, esophagus this week, or stomach wall structure next week. But pretty much the structure is the same, which means that going from the outermost to innermost, we find first serosa, then we find two muscular layers. The outermost is longitudinal muscular layer, and then the innermost is circular muscular layer. In between these two muscular layers, which are three actually in the stomach is the only part which has three muscular layers. The rest of gastrointestinal tract has two, outermost longitudinal and inner circular layer. So in between these two muscular layers, there is a nervous plexus here, myenteric or Auerbach's plexus. This is we can call it like end division of parasympathetic nervous system, or we call it enteric nervous system. There are two plexuses in the wall of gastrointestinal tract, and we call them enteric nervous system because they can function absolutely independently of any other influences. They do get influences from sympathetic and parasympathetic, but they can also work independently, and we will discuss how they do it. So after the circular muscular layer, we have submucosa, where there are glands opening into the cavity, secretory glands opening into the cavity. Between the submucosa and the circular muscular layer, there is another plexus called submucosal or Meissner's plexus. Finally, there is muscularis mucosa, mucosa muscularis, which is again a muscle layer controlled by submucosal plexus. And finally, epithelium, lamina propria and epithelial cells, which have these invaginations forming so-called brush border that, as you can see, increases the surface of gastrointestinal tract, the surface for absorption, several hundred times. Clinical case. A 36-year-old woman presents with a 3-month history of severe gastrointestinal symptoms, including persistent nausea, early satiety, abdominal bloating, and alternating constipation and diarrhea. Over the past month, she has developed orthostatic dizziness, which means that she becomes dizzy when she stands up from either sitting or lying down in bed, and dry mouth. Positive ganglionic acetylcholine receptor antibodies confirm the diagnosis of autoimmune autonomic gangliopathy. So her autonomic ganglia are being destroyed by this disease process. So we are going to discuss today where the autonomic ganglia supplying GIT are located, where do they receive sympathetic and parasympathetic input from, what is enteric nervous system, where are plexuses located, we already looked at them, and what functions they perform. When we are talking about the control of GIT functions, we are talking about two major mechanisms, nervous and hormonal. And it's important to say, and we will see it in the lectures to follow, that from oral towards the arboreal end of gastrointestinal tract, the role of nervous mechanisms decreases, while the role of endocrine increases. Thus, with regards to saliva control, for example, which is mouth cavity, there is no hormonal control, just nervous mechanisms. When we come to stomach and secretion of gastric juice, it's pretty much 50-50 nervous and hormonal. And all the way down towards the secretion of pancreas, bile excretion and production, that's pretty much hormonal control, with some input of vagus, which facilitates the hormonal influences. When we are talking about nervous system, we are talking about extrinsic and intrinsic. So extrinsic is sympathetic and parasympathetic, and intrinsic is enteric nervous system, which is found in the wall of the gastrointestinal tract. This is basically this myenteric and submucosal plexuses, which are going through all the tube of gastrointestinal tract, from esophagus all the way down to the vagus, to the anus. So autonomic nervous system has two subdivisions, parasympathetic and sympathetic. And when we talk about parasympathetic nervous supply, we say that majority of it is provided by vagus. So vagus supplies everything starting from esophagus, including transverse colon. Descending colon, sigmoid and rectum is supplied from the sacral spinal cord segments. This is S2 through S4. The ganglia are located inside the wall of the gastrointestinal tract. So parasympathetic ganglia are inside the wall. With sympathetic division, we are talking about the level of T1 through L2, so quite extensive level of spinal cord segments that are involved in supply of gastrointestinal tract. And the ganglia are paired and not paired. Paired ganglia are sympathetic chain, which is located paravertebrally on both sides of the spinal column, forming a chain, sympathetic trunk or sympathetic chain. And non-paired structures are prevertebral ganglia, like celiac ganglion, superior and inferior mesenterical ganglia. So these are sympathetic ganglia. And they send postganglionic fibers to, again, synapse on the same Auerbach's and Meissner's plexuses. So here is enteric nervous system. Let's look at this graph. So here we see Auerbach's or Meissner's plexus, which is located between two muscular layers, outermost longitudinal and inner circular muscle layers. So here we have quite an extensive collection of ganglia, which are neuronal bodies. They are also neuronal axons. We also can see here this dark blue, the fibers of vagus or sacral nerves that are synapsing on the ganglia. But apart from this, so these are ganglia and these are sympathetic, parasympathetic fibers. But apart from parasympathetic fibers, which are shown on this slide as dorsal motor nucleus of the vagus, of course they are also sacral, S2 through S4, and they are also sympathetic postganglionic. So they are preganglionic parasympathetic, synapsing on the ganglia located inside the wall of the guts, and postganglionic sympathetic, because remember the sympathetic ganglia are located outside the guts, paravertebrally and prevertebrally. All right. So this nervous system can function independently because if you look at the reflex arc, do you know what a reflex is? You know, right? Reflex arc. So it requires normally 3 neurons. In most cases, this is sensory neuron, which is yellow here. It detects signals from the mucous membrane, which may be chemical signals, mechanical stretching, they may be thermal signals, they respond to temperature of the food, they respond to chemical content of the food, and stretching. And they send these signals to the contact neurons, interneurons, which are gray here, or ganglia. And they in turn send signals to motor neurons. And when we say motor neurons, we mean that these neurons can not only control contractions. Here, this green and burgundy, they are controlling contractions because, as you can see, they are synapsing on the smooth muscles. But also in the submucosal plexus, there are efferent neurons which control secretions. So this one is also kind of motor neuron because it performs a response. It's a factory neuron, but it controls secretions. Apart from, or unlike sympathetic and parasympathetic nervous system or central nervous system, enteric nervous system employs the variety of neurotransmitters. Even the substances that are not normal neurotransmitters, like gases, for example, nitric oxide, are used, and ATP, for example, are also used as neurotransmitters by enteric nervous system. So really a lot of neurotransmitters, starting from acetylcholine to norepinephrine, to substance P, serotonin, VIP, vasointestinal inhibitory peptide, and nitric oxide, which is a gas. ATP also. So it can function independently. So what are the 2 plexuses and what do they do? So our Bach's plexus clearly controls the motor function. So it is the one responsible for peristalsis. And the fact that one neuron, afferent neuron, is connected to 2 different types of ganglionic cells and 2 different types of motor neurons, the one that are stimulatory and the one that are inhibitory, allows them to provide propulsive peristalsis when there is contraction, relaxation, and this is how the food is moving due to contraction on one end, relaxation on another, and then contraction, relaxation, and the food is moved. So apart from propulsive peristalsis, there are tonic contractions also of sphincters, maintenance of tone, and relaxation of sphincters, which is very important. And when we come to relaxation, the 2 important neurotransmitters are VIP and nitric oxide for relaxation. This is lower esophageal sphincter. We will be talking about it this week. Pyloric sphincter, ileocecal sphincter. Submucosal plexus, as you can see, can control contractions of small muscular layer, mucosa muscularis, this one, but mainly involved with secretions. It also can regulate absorption due to its effect on the vessels. Dilatation of the vessels in the mucous layer can allow for better absorption. We will still come back to the case that we had, so that's another one. A 2-day-old male newborn has not passed meconium since birth. He has progressive abdominal distension, bilious vomiting, that is vomiting with bile, and poor feeding. So the baby is crying all the time, may even look blue because of the cry, has distended abdomen, which is tympanic on percussion. You know what is tympanic sound? It's like this. So so very high. What can cause this sound? The air. Normally, if you percuss abdomen, it's not tympanic sound. But if the abdomen is distended, filled with air, that is what you hear, like a drum. Right. A rectal exam shows empty rectal vault. A withdrawal of finger produces a gush of stool and gas. No anal malformations found. Abdominal X-ray shows dilated loops of bowel with air fluid levels. A rectal biopsy, the absence of ganglion cells in the submucosal and myenteric plexuses. And this is actually the golden standard of diagnosis, rectal biopsy. Golden standard of diagnosis for Hirschsprung's disease. So what is Hirschsprung's disease? So this is how it may look like on X-ray. This is the rectum and this is sigmoid. So you can see constricted rectum and very much dilated sigmoid. So this is intraoperatively. We can see again very much dilated loops of sigmoid. Normally, so Hirschsprung disease is a congenital absence of ganglia in myenteric and Auerbach's plexuses, normally both of them, and it affects mainly rectum and sigmoid. So in the part of the gastrointestinal tract where the ganglia are absent, it remains constricted. It remains spastically constricted. It cannot relax. But the proximal segment, the segment above constriction, is dilated. Why? Because the meconium cannot pass. So the disease is usually found out within the first 48 hours of baby's birth, because babies cannot pass meconium. So this is where the problem starts. Meconium cannot be passed because the rectum stays constricted. It cannot relax due to the absence of ganglia there. So the pathogenesis of these conditions is the failure of neural crest cells to migrate into the distal column. So normally it's distal column involved from cecum to rectum. This is where we usually find aganglionic segments. So they cannot relax and they cause they cause distension of the proximal segments due to the functional obstruction at this area. There is no obstruction, right? Remember that anal exam found no abnormality. There is no actual obstruction. It's only functional due to inability of this part to relax. So we already saw the clinical presentation. The golden standard of diagnosis is rectal suction biopsy, which shows the absence of ganglia, and this confirms the diagnosis. Also, X-ray can be done. But the gold standard is biopsy. The only treatment is surgical. That's surgical resection of aganglionic segments. That's it, nothing else can help, just surgery. Talking about reflex control of functions, we say that there are short and long reflexes which are involved in the control of GIT functions. So what are these short and long reflexes? So anything that is going outside the enteric nervous system is long. So if the centers are in the cephalic brain, so if the centers are in the medulla, for example, where the nucleus of vagus is, or in the spinal cord for sympathetic nervous system, or sacral segments of parasympathetic nervous system, then the reflexes are long. So they are gray arrows on the slide. So everything going outside the enteric nervous system. with the centers being in the medulla, in the spinal cord, is already, some of them are under the control of even cortex, like for example defecation reflex. And short reflexes are reflexes that are performed within enteric nervous system. When there are local stimuli, like change of pH, change of temperature, stretch, change of osmolarity, chemical substances, products of digestion, they act on the sensory neurons, cause transmission of the signals by the interneurons to the neurons of myenteric or submucosal plexuses, and as the result there is either muscle contraction or secretion that is involved. Secretion can be exocrine or endocrine. So short and long reflexes are present, for example, in the control of gastric juice secretion, and we will be talking about both short and long reflexes found there. For saliva secretion, there are only long reflexes. It's involvement of CNS, so there is no enteric nervous system when we talk about the glands. So let's look at this case again and answer the questions. Anybody would like to start answering the questions? That's what we discussed already. So the ganglia are inside GIT and outside, right?
Yes.
Inside GIT are where? Two plexuses.
Yes.
What are they?
Submucosal and
Yes, our box is between the muscular layers.
yes. And longitude.
Submucous is where? In the submucosa. What about sympathetic ganglia
Paravertebral
and prevertebral? Right. What about? Okay. So what is enteric nervous system? What forms it? The two plexuses. Ganglia are a lot more, because they are located in both plexuses along all of the gastrointestinal tract, but two plexuses. So we already looked at them. Another clinical case. 72-year-old Caucasian female presented with persistent watery diarrhea for several weeks, accompanied by significant weight loss and muscle weakness. She reported passing stools more than 10 times per day, even during fasting periods. CT abdomen revealed pancreatic mass, and high plasma VIP level confirmed the diagnosis of VIP-oma. So every time you see the suffix oma, it means what? Tumor. Tumor. We'll be talking about gastrinoma, the tumor that produces gastrin. Now we are talking about VIP-oma, VIP-oma. So we will look at what cells produce VIP, where they are located, and whether it is endocrine, neurocrine, or paracrine agent, and what influence does it have on secretory and motor functions. So, talking about hormonal or humoral control of GIT functions, we say that it is performed by a number of peptides. So these peptides are referred to three major groups. First of all, they are classic hormones secreted by the cells of APUD system. And when we say classic hormones, we mean hormones that are released into the blood. So they are secreted into the blood, and with the bloodstream, they are carried to all the cells that have receptors to these hormones. Separate group is neurocrines. Neurocrines are substances which combine the properties of neurotransmitter and hormone because they are secreted by specific neurons, and they may be released into the usually into the extracellular fluid. Like, for example, when we are talking about the control of smooth muscle functions, if you remember what we discussed in foundations, that smooth muscles are supplied by the nervous fibers, which do not really have this definite synapse between the muscle cells and neurons, and they are
Very positive.
yes, they are very good. They are varicosities which release the neurotransmitters into the extracellular fluid, and many cells can be affected by these neurotransmitters. So this is pretty much the case, especially as we are talking about control of muscle functions, and all the muscles in the gastrointestinal tract, excluding esophagus, upper 1/3, which has striated muscles, and excluding external anal sphincter, which is also striated muscles. The rest of the muscles throughout gastrointestinal tract are all smooth. So these neurocrines are released normally into the extracellular fluid and affect muscle contractions mostly, but also can affect secretion like VIP and GRP. So GRP is a gastrin releasing peptide, which is released as a neurotransmitter by vagus, and it is causing stimulation of G cells to secrete gastrin. So it is called gastrin releasing peptide. You already know that the neurotransmitter released by most fibers of vagus, by most nervous ending, is what? Acetylcholine. But apart from this, GRP and some others can also be released by vagus. So when vagus supplies the mucous membrane of the stomach and the G cells, it's specific neurotransmitter that is released there, gastrin releasing peptide. We'll be talking about it more next week when talking about stomach, and cephalins also. Paracrines. So paracrine, when we are talking about paracrine control, we say that chemical substances are released by cells, and they are released into extracellular fluid. They cannot travel long distances in the extracellular fluid like hormones, two hormones can with blood. So they stay close by. These substances are somatostatin released by D cells and histamine. So they act on the neighboring cells. So this scheme here shows where the major hormones, the true hormones, and some of the neurocrines are released. Gastrin mainly in the antrum of the stomach, duodenum and partially jejunum. CCK, cholecystokinin, pancreatisamine is duodenum and partially jejunum ileum. Secretin GIP methylene from duodenum towards in the jejunum, and secretin ileum as well. I will stop here and we will continue from here. So there are two lectures combined. Can I answer any questions? All right. Thank you very much. You can switch off recording.